Hello my dear friends, welcome to Live Prep. In the previous lecture, we studied about the factors affecting the enzyme activity. Now this is the last lecture of the biomolecules and where we are studying about the classification and nomenclature of enzymes, and also we are studying about the cofactors. And these are all very small and clear-cut direct information. So we don't need to spend much time on this. And at the end we will also read the summary. So there are no new points in the summary, just I will read it. Okay, so let us try to quickly complete this particular part. We don't need to worry too much about this classification of the enzymes and the nomenclature. So thousands of enzymes were discovered by the scientists, but the thing is we need to give some names for this. So what there is a group of scientists, and it is called as enzyme commission, and then they have decided some rules. See, generally we are studying about the nomenclature in the case of organic chemistry, in the case of botany, zoology, likewise it is there, no? In the same way, for the enzymes also there is a commission. It is called EC, enzyme commission. So this enzyme commission, it has referred a four letter, so 4-digit numbering for that each enzyme. For every enzyme there will be 4-digit like this. So in these digits, so what actually they represents? The first digit represents the main class. So what is the class to which they belong? Second one will represent subclass, third one will represent subclass, and the fourth one is actually a serial number. So in that, within that subclass, so for different enzymes, so they have given the different numbering simply for the differentiation. So in this way they have mentioned one 4-digit ID for each and every enzyme. So in this way, at our syllabus, in our NEET syllabus, for NEET UG syllabus, we are studying only on these classes. That's it. We are not talking about subclass, subclass and serial number, but you should know that 4 digits are present in this. But we are focusing only on the first one, that is the class. That's it. In that class also, simply we are writing the names of this and what happens in single sentence. That's it. We are not studying any further examples, because unnecessarily if we are asking some questions unnecessarily on this, so again you will be confused. So don't worry about this. Simply NCERT sentences, names of these classes and the sentences, that is enough. Suppose they are giving one example and to which class it will belong, they can ask like this, but if you understand the concept of these particular words, you can easily understand that particular one. But we are focusing only on the first one, so please remember this. So in this way, so 6 classes are there, and in each class again, depending on the class size, again 4 to 12 different subclasses are there based on the categories and all. So in this way they have given this particular one. Clear? And here enzyme commission has given this, that's why EC will be there, and then 4 digits will be there. First one is class, second one subclass, third one subclass, last one serial number, and we are focusing only on the first one. And here also there is a fixed sequence. You need to remember the sequence. What is the sequence? O, T, H, L, I, L. This is the first letters I wrote here. Okay. And please remember, oxidoreductases, it is fixed 1. Suppose here they are giving 1. 1 means what is this one? Oxidoreductases. That means this enzyme will belong to which class? Oxidoreductases. Suppose instead of 1, if number 2 is there, number 2 is what? Transferases. You need to remember in the same sequence. Suppose here 3 is there, it is hydrolases. 4 is present here, it is liases. If 5 is present, isomerases. 6 is present, ligase. That's it. In this way only 6 will be there. So in this way, sometimes what they will do, to confuse us, they will give that. So EC 4.2.1.5 they will give, and it belongs to which class? So you don't need to worry about this. So here they give 4. So 4, number 4. So what is that number 4? It is liases. So in this way, you just focus on only the first digit. In the first digit, only 1 to 6 numbers will be there. So according to this number, you should write it. So I have written this first letters, observe carefully. Oxidoreductases, transferases, hydrolases. L is coming 2 times, but hydrolases will help in breakdown of the bonds with the involvement of water. But in the case of liases, without involvement of water, rearrangements will occur. So these two are related. So here hydrolases, this is liases. Isomerase, last one is the ligases. Simply we will understand directly from the NCERT. Directly, just single sentences they give. You don't need to worry too much about these things. Now let us quickly go through this. Thousands of enzymes have been discovered, isolated, and even studied also. Most of these enzymes have been classified into different groups based on the type of reactions they catalyze. So different enzymes are catalyzing different reactions. That's why different classes, different groups are present. Enzymes are divided into 6 classes. What are these 6? With names, numbers you should remember. This is compulsory. And each with 4 to 13 subclasses. Subclass you don't need to remember. Simply remember the sentence. That is enough. And named accordingly by 4-digit number. So 4 digits are present in number of each and every enzyme. Next one. What is the first one? Class number 1. Class number 1 is oxidoreductases. Oxidation reactions will occur. Not only this, dehydrogenase. What is dehydrogenation? D means removal. Removal of hydrogen. Removal of hydrogen means it is oxidation. So again oxidation reduction only. So in this way, any of this name you can use it. So here what happens? Oxidation process, reduction process. So these are the things that are happening here. The enzymes that are involving in oxidation and reduction of some of the substrates. So now this type of, that type of enzyme will come under this particular group. This is what they are saying. Enzymes which catalyze oxidoreduction between two substrates. That means one is oxidized, other one is reduced. This type of reactions will occur. Clear? Second number, class number 2, transferases. They will transfer some groups other than hydrogen. If it is hydrogen, again it will come under oxidoreductases. Other than hydrogen, some groups will be transferred. So that is called transferases. Okay. So transacylases or acyl transferases. They will transfer acyl group. So in this way you can understand this. So enzymes catalyzing transfer of a group G, that should be other than hydrogen, between a pair of substrates. So from this already a group is attached to this. Now it is donating its group to some other molecule. So that now these are the products that are produced. In this way, other than hydrogen, if some groups are transferred, now they are called transferases. Those enzymes will come under transferases class. That is the second class. Now let us go through third class, hydrolases. Hydro, water. So with the involvement of water, bonds are broken here. With the involvement of water, that means water molecule is used while breaking this particular bond. So in this way, all these reactions will come here. So enzymes catalyzing the hydrolysis of ester bonds, ether bonds, peptide bonds, glycosidic bonds, C bonds, C halide bonds, PN bonds, wherever the bonds are broken with the involvement of water, all of them will come under hydrolases. Yes or no? In simple, if you are talking all the digestive enzymes, they are using the water molecule for breakdown of so many bonds. So all these are various examples. All these are examples for hydrolase. So many examples we will study for hydrolases. Okay. Next one, liases. Liases, what they are doing, without involvement of water, some groups will be removed, so that generally they are involving in the formation of carbon-carbon double bonds. So here X is there, Y is there. Now these two are removed and carbon-carbon double bonds are formed. But in this case, the important point is water molecule is not used in during this particular one. Enzymes that catalyze removal of groups from substrates by mechanisms other than hydrolysis, so that they are ultimately leaving double bonds between the carbons here. So in this way, if the enzymes are involved, they will belong to this liases group. And the next one is regarding the fifth class. Class number 5 is isomerases. Different isomers in the chemistry you will study about positional isomers, geometrical isomers, likewise many isomers we are studying. Any of these, so from one form to other form, if it is converted, so all of them will come under isomerases. So includes all the enzymes catalyzing interconversion of optical, geometric, positional isomers, any of these. If one form is converted to other isomer, so they will come under isomerases class. Next one, last class number 6. That is the ligase, ligation. Please understand the meaning of this word ligation, because in biotechnology also we will study this. Ligation means attachment. So now we understood the meaning. The enzymes that are involving in attachment of some components, that means some attachment of some compounds, they are called as, those enzymes will come under ligases group. Enzymes catalyzing the linking together of two compounds, like enzymes which catalyze the joining of CO, CS, CN, PO, etc. Likewise, they are attaching so that they are forming this type of bonds. Any of this, in this way, if two compounds are attached, in that process, if enzyme is involved, that enzyme will belong to this ligases group. That's it. So in this way, only 6 classes are there. Remember the class name in the sequence. In the same sequence, you just remember. Simply remember the function. That's it. You don't need to worry too much about this. Whenever they are asking the questions, directly they are giving these statements only. That's it. So you don't need to worry too much about this. And with this part is completed. And finally, we are going with the last concept called cofactors. For this cofactors, anyhow, I have given this particular flow chart, but better I will write it here. So generally the enzymes, Generally the enzymes. So some enzymes are having single protein, some enzymes are having multi subunit, whatever it is. So in this way different enzymes are present. But here we are talking about some enzymes, they are having only protein part. Only protein part. Some enzymes are having only protein part. That means just protein itself is acting as an enzyme. Protein part is there. But in some cases, in some enzymes, protein part is present. Protein part is present plus a non-protein part is also present. Please remember this. Protein part and non-protein part. In this way, two parts are present. Some enzymes are having only protein part, that's it. But some are having non-protein part along with the protein part. So now here we are giving the names. Those enzymes that are having only the protein part as an enzyme, so they are called simple enzymes. So simple enzymes are having just protein as the active molecule. So that itself is doing the reaction. But here we are using a word called conjugate enzymes. Conjugate enzymes. And in the case of conjugate enzymes, what we are saying? Protein part, non-protein part, two are there. What is this protein part is called as? Protein part is called as apoenzyme. Protein part is called as apoenzyme. And this non-protein part is called as cofactor. Non-protein part is called as cofactor. This is the same thing that they have mentioned here. Enzymes are classified as simple enzymes and conjugate enzymes. In the simple enzymes, only the protein part is there. But in the conjugate, protein part, non-protein part, both of them are present. So the protein part of conjugate enzyme is called as apoenzyme. Non-protein part of conjugate enzyme is called a cofactor. Please remember here, in this case only protein part is there, but it is functional. It is functional. The only protein part of conjugate enzyme is called apoenzyme. It is not functional. Please remember, apoenzyme on its own, it cannot do the reaction. It cannot catalyze the reaction. Compulsorily it needs cofactor because it is conjugate enzyme. But here cofactor concept is not there. It itself can perform this one. So please remember, apoenzyme. So normal simple enzyme can work on its own, but apoenzyme cannot function on its own. Compulsorily it should interact with cofactor. Now what are these cofactors? Simple point here. Cofactors. In NCERT directly they are talking about three types of cofactors. So what are these? Prosthetic group, coenzyme, and then metal ion cofactor. Three names they have given, but what I am doing is, before directly going into these three, let us first basically classify into two types. Some are called as organic cofactors, some are called as inorganic cofactors. Inorganic cofactors means generally the metal ions will act as cofactors here. But in the case of organic cofactors, again two types are present. Some types of organic cofactors are tightly bound with apoenzyme. Protein part is called apoenzyme. To the apoenzyme, it is tightly bound. What is the meaning of tightly bound? It is always attached. To the apoenzyme, it is tightly bound and always it is present. And that type of cofactor is called as a prosthetic group. That is called as prosthetic group. But there are some other types of organic cofactors, but they are transiently, what is the meaning of transiently? Temporarily attached or loosely attached, loosely bound, transiently attached, temporarily attached. What is the meaning of this loosely or temporarily attached? Whenever there is a reaction, please understand the meaning of this. Whenever there is a reaction, then they will come and attach to apoenzyme. They will do the reaction. Once reaction is completed, they will go away. Understood or not? But the prosthetic group, it is always attached to apoenzyme. But the coenzyme, it is the organic cofactor, but it is loosely or temporarily or transiently attached to apoenzyme. It will come only when there is an requirement for reaction. When there is a reaction, then only it will come and attach, and it will perform the reaction, and then it will go away. So that's why they are loosely attached, but prosthetic group is tightly attached. But these also, so they also come, they can come and go, but they will come under inorganic cofactors. In this way, cofactors are three types: prosthetic groups, and then coenzyme, and then metal ion cofactor. In the NCERT, they have given some examples here. Prosthetic group example they have given heme. Heme is the prosthetic group for peroxidase and catalase. What this peroxidase and catalase will do? So already in the cell chapter itself we studied this reaction, right? So H2O2 is converted into H2O plus O2 is converted into H2 and O2. This is what the reaction. In this reaction, catalase and peroxidase are used, and in those enzymes, this heme group is the porosity group. This is compulsory. If heme group is not present, they cannot work. So that's why it is compulsory. That is the cofactor. And then regarding the coenzyme, it is temporarily. Whenever there is a requirement, they will come and do the reaction, and then they will go. And then generally these coenzymes are produced from the vitamins. Generally, they are produced from the vitamins. In the NCERT, they are talking about NAD, NADP, NAD and NADP. Nicotinamide, adenine dinucleotide. It is NAD. This is what. What is NADP? Nicotinamide adenine dinucleotide phosphate. Like this. So in this way, NAD, NADP, these are the two examples given in NCERT. These are considered as coenzymes. And from where they are produced? Nicotinamide, nicotinic acid. Nicotinic acid is nothing but niacin. This is a vitamin B complex vitamin. Yes or no? It is part of vitamin B complex. Yes or no? So generally these coenzymes are produced from the vitamins. So NAD, NADP are produced from the vitamin called niacin. It is also called nicotinic acid. Even though not mentioned in NCERT, generally you will study in the next coming chapters. FAD, FADH2. Likewise, you will study, right? FAD, FADH2. In this FADH, you just observe this case. What is this FAD? Flavin, adenine dinucleotide. This flavin means from which vitamin it will be produced? From riboflavin. From the riboflavin it will be produced. So in this way, there are different types of coenzymes are present. Generally, they are produced from different types of vitamins. That's what they are saying. And regarding the metal ion cofactor, one example they give. You need to remember this. Zinc is the important cofactor for the activity of this carboxypeptidase. Carboxypeptidase, what is the action of this particular one? It will help in the breakdown of the proteins from the C-terminus end. From the C-terminus end, 1,1,1,1 amino acid it will remove. So in this way, it is called a proteolytic. Proteo means protein, lytic means breakdown, protein breaking. So it is a protein digesting enzyme. So carboxypeptidase is the protein digesting enzyme. For the activity of this, it needs a metal ion cofactor. In this case, it requires zinc, Zn+2. Zn+2 it requires. These are the examples. With this part is completed. Now let us quickly go through NCERT reading part. Cofactors. Enzymes are composed of one or several polypeptides. So some enzymes are having single polypeptide, some are having many. However, there are a number of cases in which non-protein constituents called cofactors are bound to the enzyme to make the enzyme catalytically active. In some cases, what they are saying, anyhow protein part is there. Along with the protein part, some non-protein part is also present for the activity of the enzyme. And in these instances, the protein portion of enzyme is called as apoenzyme. So if protein, non-protein two are there, the protein part is called as apoenzyme. And three types of cofactors are identified. They are porosity group, coenzymes, and metal ion. These are the three important types. And what are porosity groups? These are organic compounds that are distinguished from other cofactors, that they are tightly bound to apoenzyme. This is very important. Porosity group, organic compound, and it is tightly bound to apoenzyme. Very important point. Next one. For example, in peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen. So what these enzymes are doing, they are converting that H2O2 into H2O and water and oxygen, right? So I think the same thing I wrote. Or did I write something else? Yes. Okay. So water and oxygen. And in this case, heme is the porosity group and it is part of the active site of the enzyme. So in this way, this heme group is compulsory. Without the heme group, they cannot work. That's why it is called as cofactor. Next one, coenzymes are also organic compounds. These are also organic compounds, but their association with apoenzyme is only transient. That means it is temporarily. That means usually occurring during the course of the catalysis. That means whenever there is a reaction, catalytic reaction is taking place, only at that time it will come and attach to apoenzyme and it will perform the reaction. Once reaction is completed, it will be removed. That's why it is temporarily attached, loosely attached, you can say. Furthermore, coenzymes serve as cofactors in a number of different enzyme catalyzed reactions. In different types of reactions, they are acting as cofactors. The essential chemical components of many coenzymes are vitamins. Majority of these coenzymes are derived from vitamins. Example, coenzyme called nicotinamide adenine dinucleotide, NAD, nicotinamide adenine dinucleotide phosphate, and all these are containing the vitamin called niacin or nicotinic acid. Niacin is also called nicotinic acid. In this way, FAD, FADH2, if you are talking about this, so now they are also FAD, FADP, all these are there. So they are produced from riboflavin, that is also a vitamin. So that's what they are saying. Next one. A number of enzymes require metal ions for their activity, which form coordination bonds with side chains at the active site, and at the same time form one or more coordination bonds with the substrate. So some of the metal ions, what they are doing, on one side they are interacting with active site of enzyme, active site of enzyme, other side they are interacting with substrate. So in this way they are very important for the particular reaction. That's what they are saying. Sorry. Zinc is cofactor for the proteolytic enzyme carboxypeptidase. Zinc is very important for the activity of carboxypeptidase, which is a proteolytic enzyme, protein digesting enzyme. In the digestion chapter, I will tell you about this name, even though the chapter is deleted. Many times we are studying these names, at least some important names I will tell you. Okay, next one. Catalytic activity is lost when the cofactor is removed. That means only apo enzyme cannot function. If this cofactor is removed, catalytic activity will be lost from the enzyme, which testifies that they play very crucial role in the catalytic activity of the enzyme. That means if cofactor is not present, apo enzyme cannot function on its own. That means they are very important for the activity of enzyme. Now we will quickly go through the summary. Nothing to explain here, simply I will read it. Although there is a bewildering diversity of organisms, so different organisms are there, so that we are confused ourselves, right? So many different types of organisms are present. Their chemical composition and metabolic reactions appear to be remarkably similar. Even though different types of organisms are present, almost all the chemical components are present in all the organisms. Next one. The elemental composition of living tissues and non-living matter appear also to be similar when analyzed qualitatively. So what is present, what is not present? In this way, qualitatively, if you observe living and non-living things, both of them are having almost the same list of compounds. But however, a closer examination reveals that the relative abundance of carbon, hydrogen and oxygen is higher in living systems when compared to the inanimate matter. When compared with non-living things, if you are observing in the living organisms, carbon, hydrogen and oxygen are higher. In the regular content, they mentioned only carbon, hydrogen. And here they are talking about hydrogen also. Anyhow, that comparison I already told you, so go through that comparison. The most abundant chemical in the living organisms is the water. 70 to 90% is the water. Next one. There are thousands of small molecular weight compounds which are having less than 1000 daltons, and they are the biomolecules. So many different types of biomolecules are present. Amino acids, monosaccharides, disaccharides, sugars, fatty acids, glycerol, nucleotides, nucleosides, nitrogenous bases are some of the organic compounds seen in the living organism. So many types of organic compounds are present. Some examples they gave here. There are 20 different types of amino acids and 5 types of nucleotides are present. Already we studied. Fats and oils are glycerides, in which fatty acids are esterified to glycerol. So in the case of these fats, mainly what happens? Fatty acid is attached to this glycerol. Yes or no? So in the mainly in the lipids we are observing. Phospholipids contain, in addition, a phosphorylated nitrogenous compound. So along with this, in phospholipids, one phosphate group is also present. That's what they are saying. Only 3 types of macromolecules, only 3, not 4. Lipid is not considered as macromolecule. Only 3 types of macromolecules are present. Proteins, nucleic acids and polysaccharides are found in the living systems. Lipids, because of their association with the membranes, separate in the macromolecular fraction. So originally they are small molecules, but since they are forming the vesicles, they are coming in the macromolecular fraction. Biomacromolecules are polymers. So first 3, they are the polymers. They are made up of building blocks which are different. Different macromolecules are made up of different building blocks. Proteins are heteropolymers made up of amino acids. Nucleic acids, DNA and RNA are composed of nucleotides. Biomacromolecules have a hierarchy of structures. Different levels of conformations are present. Primary, secondary, tertiary and quaternary. Nucleic acids serve as genetic material. The major function of nucleic acids is genetic material. Polysaccharides are components of cell walls of plants, fungi and also even the exoskeleton of the arthropods. They are also acting as some of the polysaccharides, they are having the storage forms of energy like starch and the glycogen. Proteins serve a variety of cellular functions, different functions. Already the table we studied. Many of them are enzymes, some of them are antibodies, some are receptors, some are hormones, and some others are structural proteins. Structural proteins means they are helpful for formation of some structures. Collagen is the most abundant protein in the animal world, whereas Rubisco is the most abundant protein in the whole of the biosphere. In the entire earth, that is the most abundant protein. Enzymes are the proteins which catalyze the biochemical reactions in the cells. So majority of the majority of the enzymes are proteins. Ribozymes are the nucleic acids with catalytic power. Ribozymes are the enzymes, but they are nucleic acids. Catalytic power means what? Enzyme activity, but it is a nucleic acid. Proteinaceous enzymes exhibit substrate specificity. So different proteinaceous enzymes. So enzymes made up of protein, they are having substrate specificity. That means specific enzyme is always binding with specific substrate, and they require optimum temperature and pH for the maximum activity. And they are denatured at high temperature. At high temperature, they will be denatured. Enzymes lower the activation energy of the reaction. Please remember, enzyme is not increasing the energy of substrate. It is lowering the activation energy and enhance greatly the rate of the reaction. In this way, it is increasing the rate of reaction. And nucleic acids carry hereditary information. Nucleic acids like DNA, they are storing the information for next generation. That's why they are storing the hereditary information and are passed on from parental generation to the progeny. So through the gametes, when chromosomes are going into the gametes, now they are also going into the gametes. And upon the fertilization of the gametes, now this DNA is going into the next generation. In this way, nucleic acids are moving from one generation to next generation. So since they are carrying the genetic information, so they are deciding the characters in the next generation. That's it. This is the overall information related to biomolecules. With this, it is completed. Thank you.